you go.
Okay. Thank you very much. My name is Phat Dang. I work at the National Peanut Research Laboratory looking at drought tolerance using mostly rainfall shelters, but sometimes we'll have collaborations in the field with Auburn University to look at dryland situation. So in collaboration, we have Ron Sorensen and Marshall Lamb as a co-author at the National Peanut Research Lab. And we also have collaboration with Jinesh Patel and Charles Chen on this research. My title is going to be the phenotyping of the U.S. mini-core collection to identify tolerant peanut genotypes using environmental control rainfall shelter. So when we look at drought, the reason why we say is that there's probably a very high percentage of peanuts that's acreage that's non-irrigated. And because of competition of water and the temperature, that there's going to be probably a higher percentage of peanuts that could be grown without irrigation. So the effect of biological is that it affects germination. Farmers target early planting for this reason to see if they can capture the moisture that's left over by rainfall during the winter season to germinate. Under irrigation conditions, you can probably put out water if you have water to get the growth going. Of course, growth is important. The plants need that water to grow until they're about full canopy coverage, which is about 60 days after planting. And then, of course, it affects yield and quality. So, in some aspects, drought is maybe related to pre-harvest aflatoxin. So there are several mechanisms that are thought to be adaptive for plants. One is the escape or avoidance mechanism, where the plants try to germinate quickly and then grow as fast as they can and then finish. And so that's really more of a survival mechanism to maybe perhaps get the seeds out, and because of year after year extreme situation, plants want to survive, and usually it's associated with dormancy or quiescent states after the seeds have start to imbibe. The other mechanism is resistance, and that's basically plants that are grown in a very arid or semi-arid conditions year after year, and it's maintained to avoid or delay water loss quickly, shut down its growth, and maintain some sort of survival mechanism, produce flowers quickly, and regardless of yield, and then to maintain metabolic pressure process until the whole process is done. It's very similar in a sense to escape or avoidance, but it also is a way for plants to produce as much seeds as it can so that it becomes a competitive advantage. A lot of the plants that we deal with in terms of the cultivated peanuts is more of a tolerance mechanism, where there you have some osmotic adjustments and stability to the membrane stability in terms of enzymes or perhaps lipid or changes in the types of osmotic potential like sugar. Eventually oxidative repairs and then transport system, and then of course gene expression to produce long-term effect for tolerance. So the advantage of that is that tolerance actually the plants maintain a very high transpiration efficiency to produce as much seed and quality as possible, and that's what we're targeting. So when we first started doing this work, we wanted to know if there's a yield correlation with the certain developmental stages of drought tolerance, and we want to develop a sort of like a strategy to evaluate genotypes, different genotypes in the Southeast area. So we did a 21-day drought stress on these things, and we picked an early season, which starts around 30 days after planting, the middle season, which is around 60 to 70 days, and then the late season, which is about 90 to 100 days after planting. This is target to see if there is a yield correlation. Again, the advantage of using the rainfall shelter is that once the rain comes, even a few drops would trigger a closure, and it'll stay closed for about 30 minutes, then open up again. In terms of irrigation control, we can apply measurements to make sure that the plants get what it needs. Based on an 8-inch and a 12-inch depth of measure, a moisture measure and temperature. And so at minus 40 kilopascal with the average of between the 8-inch and a 12-inch, we'll start a trigger irrigation for irrigation control. So when we started this work, we chose 5 different genotypes. 2 was provided by Dr. Corley Holbrook, the C7616 and A104, that he thought was very has the capacity to have drought tolerance. We added Georgia green because we saw that there was a little bit of difference mechanism. We find that it accumulates a bit of sugar, and that farmers, when they grow that in many years, if there is a drought that occurs, Georgia green would just sit there until rainfall comes, and then it'll start growing again. So it has that capacity. And we've added AP3 as a susceptible line for both yield and physiology. And then another line, O0 T12, was identified by Charles Chen that had minimal water requirement in Texas. So when we look at the different early stages yield of all these 5 genotypes, we find that 30 days really has kind of a minimal yield loss, even though there's about 21 days or 30 days of drought, then followed by full irrigation. The mid-season, which is around 60 days again, has significant yield loss, as much as 50% in one of this genotype, and then at least 25% yield loss compared to full irrigation and late season, as well as rainfall treatment. So our objective really, again, is to look and see if we can identify genotypes that will have the mid-season drought tolerance. Again, really balancing the effect of the yield production to its recognition of drought and respond to it. So we evaluated 162 peanut accessions, including 120 US mini-core that was been evaluated by our group for many of the traits. We wanted to associate the physiological measurements, and we used surrogate measurements of relative water content and specific leaf area, and we added the leaf dry matter content to see if there's other chemical or sugars that are produced to maintain the drought stability under yield to performance. We applied this to the GWAS on the US mini-core to identify gene candidate drought genes. So in the mid-season drought, we wanted to do a 21 days to up to 30 days of stress. When we start, stage 1 is when we have no stress on it, but it's beginning of our test. Stage 2 is 1 week after stress, and so forth, 3, 4, until about the 3rd week, and then we irrigate and we do a measurement for recovery, and then it's irrigated for the rest of the treatment. So this work was done experiment was conducted at the National Peanut Research Lab in Dawson, Georgia for 2017 and 2018. It's 2 reps, about 5 feet blocks, or 1 meter row reps. That's done two randomized complete block design. With the best agronomic practices, we've irrigated to make sure the germination occurs evenly. Then for the we'll irrigate until the start of the treatment, and then we'll stop irrigating and start the progressive drought. Once the drought is over, we'll water and come back to regular growing application. Also, fungicide and all kinds of other agronomic treatments are done based on recommendation. For the relative water content, leaf specific area and physiological measurements, we collected 3 fully expanded leaf from the main stem of the subplots. Then for the drought rating, we waited until about 3 or 4 weeks until we can see somewhat difference between the genotypes. And we use a scale of 1 through 5, where you have 1 being kind of a mild response, very limited wilting, and 5 being a severe drought symptoms. And we did target around 3 p.m. for the hottest part of the day. For yield, dry pod weight, and pod number, we waited until after curing to do these measurements. The plots were inverted at around 140 days and picked with a Australian picker. So this shows an overhead picture of our plots around 3 weeks after the drought. You can see that there's some variations across different plots. So for the surrogate trait water use efficiency, we use relative water content. That's a fresh weight minus dry weight over the turgid weight minus the dry weight. So the fresh weight is obviously when we harvest. For the dry weight, we put the plants into the heat incubator for 60 degrees for about 10 days, and we'll take it out and measure it. For the turgid weight, after we do the full weight, fresh weight measurement, we'll put it in water under light condition for at least 3-4 hours. Then we'll take that out and blot it dry, and then do a measurement of weight. For specific leaf area, we did a leaf area measure, also divided by the leaf dry weight. So leaf dry matter content is leaf dry weight over leaf turgid weight. So when looking at variance component of variability, we divided up into the physiological traits, relative water content, specific leaf area, and leaf dry matter content. What we see is at 1 week after the drought, we see there's a bump in heritability from at week 1 after drought, but then it significantly drops off, and where we really become insignificant at this point. There are no differences using the specific leaf area, even at 1 week after the beginning of drought, and then no differences later on at any of the other stages. For the dry leaf dry matter content, we see no differences here, so we can't really use these two measurements for drought selection. Using variance components for heritability, use measurement of dry weight, pod count, and drought rating. We can see for the variance of environment, genetics, genetic and by environment error, we can see that heritability is around almost 70% for dry weight. Pod weight, pod count, we can see up to 66%, and drought rating is around 28-29%. This looks at the frequency distribution between 2017 and 2018 for both dry weight, dry pod count, and drought rating. What we can see is there is a difference between the 2 years. If you look at 2017, we have a very dynamic range between 0 and 350 grams pods for some of the genotypes, but in 2018 we only see the maximum about 200. The 2
minutes, Phat.
Okay, normal distribution for this and very correlated to the pod count. The drought rating for 2017 was heavily towards the more severe terms, but for the 2018 is more randomly distributed. So these are the genotypes that was ranked 1 through 8, the best rankings based on both pod count, pound dry weight, and then the pod unit. So these are relative terms, but it's the best, and we can use this in the future to validate this situation. We applied a GWAS genome-wide association analysis where we used 120 part of the mini-core collection that was been characterized by our group. And then we used the genotype mix array developed by Clevenger. We used the phenotyping pod count, yield, and drought rating. And we use a GAPIT, the R the R suite for linear model, mixed model, and then the combination of the fixed and random models. So our results, this shows a distribution of Snips that's characterized by Clevenger. We looked at the GWAS analysis. If you look at the left, we see a correlation expected log to the observed. You can see a number of Snips that are above this level. Break log of greater than 0.0003, significant for these Snips. Again, there's fewer for 2018, but there's some Snips that are variable. In terms of pod count, we only see one significant SNP for 2017, but we see a lot more. Again, you see differences in the environmental effects of 2017 to 2018. In terms of drought rating, we really see no SNP differences in the drought rating, and it's difficult because it's more subjective. And when we do the drought rating, probably the peanut plants have been recognizing, and it's switching from a short-term drought recognition to a long-term drought response. So this is a summary of significant SNPs that we've seen for both dry weight and pod count. We see that a number of them for 2017 and 2018 both have the positive or negative correlation. So candidate genes were found around these 100 kilobases of affinity to these markers. And for the dry weight in 2017, we identified on chromosome 8, where you see different types of genes that are there, F-box, nuclear factors, et cetera. For dry weight number 2018, we see a number of genes that are different, sucrose, synthase, ATP transporter, and stuff like that. For the pod count 2017, we see the main, membrane transporters, ATP dependent helicases, auxin-like proteins. And for pod count 2018, we see some of transcriptional factors. So the point here is that there are really differences in the 2 years that we evaluated. And so the yield component is a composite of what was recognized before by the peanut plant, the duration of the drought that the plant has occurred, to give you a yield component. So again, you can see the numbers of a different pod number as well. So in our future research, we want to develop primers to these newly identified SNPs to validate this whole thing, and to genotype and to do more of the field phenotyping, both in the rainout shelters and in the dryland situation, and apply it to the drought, these drought regulated markers in breeding programs. So thank you.
Thank you, Phat. I think kind of maybe in an interest of time, we'll have to move on to the next presenter. If you do have any questions based off of Phat's presentation, can you please enter them into the Q&A session, and we'll make sure that he is able to answer them directly to you. Thanks again, Phat.
Thank you.